Chapter NO10 Microbes in Human Welfare. Microbes in household products. Lactobacillus and others commonly called lactic acid bacteria lab grow in milk and convert it to curd. During growth, the lab produces acids that coagulate and partially digest the milk proteins. It also improves its nutritional quality by increasing vitamin B12. Lab play very beneficial role in checking disease-causing microbes in stomach. The dough, which is used for making bread, is fermented by using baker's yeast Saccharomyces cerevisiae. Toddy, a traditional drink of some parts of southern India, is made by fermenting sap from plants. Cheese is one of the oldest food items in which microbes were used. The large holes in Swiss cheese are due to production of a large amount of CO2 by a bacterium named Propionibacterium shermani. The Roquefort cheese is ripened by growing a specific fungus on them for a particular flavor. Microbes in industrial products. Production on an industrial scale requires growing microbes in very large vessels called fermenters. Are fermented beverages. The yeast Saccharomyces cerevisiae used for bread making and commonly called brewer's yeast is used for fermenting malted cereals and fruit juices to produce ethanol. Wine and beer are produced without distillation, whereas whiskey, brandy, and rum are produced by distillation of the fermented broth. The antibiotics. The antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease-causing microbes. Penicillin was the first antibiotic to be discovered and it was a chance discovery. Alexander Fleming White, working on Staphylococci bacteria, once observed a mold growing in one of his unwashed culture plates around which Staphylococci could not grow. He found out that it was due to a chemical produced by the mold and he named it penicillin after the mold Penicillium notatum. Later, Ernest Chain and Howard Florey made its full potential effective antibiotic. See, chemicals, enzymes and other bioactive molecules, chemicals Aspergillus niger, fungus, citric acid Acetobacter aceti, bacterium, acetic acid Clostridium butylicum, bacterium, butyric acid Lactobacillus, bacterium, lactic acid Saccharomyces cerevisiae, ethanol. Enzymes, lipase used in laundry detergents, pectinase and protease used in bottled juices, streptokinase, Streptococcus bacterium used as clot buster to remove clots. Bioactive molecules, cyclosporin, Trichoderma polysporum fungi used as immunosuppressive agent for organ transplant patients. Statins, Monascus purpureus yeast used as blood cholesterol lowering agents. Microbes in sewage treatment. Treatment of wastewater is done by heterotrophic microbes naturally present in the sewage. This treatment is carried out in two stages. Primary treatment or physical treatment. It involves physical removal of particles from the sewage through filtration and sedimentation. Sequential filtration to remove floating debris, sedimentation to remove grit, salt and small pebbles, all solids that settle form the primary sludge, and the supernatant forms the effluent. The effluent from the primary settling tank is taken for secondary treatment. Secondary treatment bata biological treatment. The primary effluent is passed into large aeration tanks. This allows vigorous growth of aerobic microbes into flocks. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the BOD biochemical oxygen demand of the effluent. BOD is a measure of the organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. Once the BOD of sewage water is reduced significantly, the effluent is then passed into a settling tank where the bacterial flocks are allowed to sediment. This sediment is called activated sludge. A small part of this sludge is pumped back into the aeration tank to serve as the inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digesters. During this digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. These gases form biogas. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams. Microbes in production of biogas. Biogas is mixture of gases produced by the microbial activity and which may be used as fuel. Certain methanobacterium present in gut of ruminants which grow anaerobically on cellulosic material produce large amounts of methane along with CO2 and H2. A lot of cellulosic material present in the food of cattle is also present in the rumen. Cellulose is broken down and converted later into biogas. Thus, the excreta dung of cattle, commonly called gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called gobar gas. Biogas plant. The biogas plant consists of a concrete tank in which bio waste are collected and slurry of dung is spread. A floating cover is placed over the slurry, which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. The spent slurry is removed through another outlet and may be used as fertilizer. The biogas thus produced is used for cooking and lighting. Microbes as biocontrol agents. Biological control of pests and diseases. Ladybird to control aphids, dragonfly to control mosquitoes, Bacillus thuringiensis, BT cotton to control wide range insects, Trichoderma, fungi, protects root system and control plant pathogens. Baculoviruses, nucleopolyhedrovirus to attack insects and other arthropods. Microbes as biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi and cyanobacteria. Bacteria, symbiotic bacteria, rhizobium with root nodules of leguminous plants free living in the soil, Azotobacter and Azospirilla. Fungi, symbiosis, mycorrhiza with root system of genus Lomus and absorb phosphorus and water from the soil for the plant growth. Cyanobacteria, symbiosis, Anbina in Azona free living, Nostoc, Oscillatoria and blue-green algae. Some